Okay, good morning. I hope everybody had a nice spring break. I'm sure all of you feel the same way that I do, that it was nice, but not long enough, right? Um, so as we kind of get back into our classes, a couple things I wanted to mention. So your TIs have completed grading the exam that we took before spring break. So your current grades are displayed in La Lima. So you should be able to check and make sure that your current standing in the class is what you sort of expect, and there's no surprises there. At this point, if any of you are concerned about where you are in the class, now that we've had a couple of exams, reach out to me, let me know, and I'll be happy to set up a time when you and I can talk about it. Okay. The other thing I wanted to mention before I forgot is that this Friday is Kuhio Day, so it's a holiday, no class. And next Friday is Good Friday, so also no class. Right? So both of these weeks are shorter weeks. Now, for those of you who have recitations on Fridays, this means you're not going to be able to meet at your normal Friday times for recitation this week or next week. So what we will do is your TIs for the Friday section students, they will give you a schedule of when all of the Thursday recitation sections meet over Zoom. And you will have the ability to go ahead and choose a Thursday recitation this week and next week to attend instead. Right. I think for most of you who are affected by this, you should be able to find a Thursday section that you can kind of just join. And for those of you who cannot, because none of the Thursday sections work with your schedule, let your TI know. And that way we can kind of keep track as to who's able to make a recitation and who cannot. All right, so keep an eye open for that. Again, this only applies to the Friday recitation students. All right. Let's move on in our discussion and talk about the role of energy inside of our cells. Now, throughout the semester, we have certainly talked about energy when we talk about things like bonds that are often at higher energy states where the electrons are in orbits in the surrounding, nuclei that might have higher energy potential. And we said that the breaking of bonds and the reforming of bonds often takes place when you have better, lower energy electron orbitals for those electrons to be able to participate in. And we said that that's really a key way of thinking about how biological molecular processes happen. Because after all, biochemistry, the stuff that's happening inside of your cells, is all about the making and breaking of bonds. Right? And so inherently, as part of that discussion, is this idea of energy and how energy is utilized by the cell, right, to be able to drive all of these biological functions. So let's start off today with kind of an introduction to energy and thinking about energy in living systems. So I grew up on the East Coast, and some of the fondest memories I have are walking through the woods, in spring. And this is after like a long cold winter when everybody's like tired of the snow and the ice. And we finally start having some sunshine and it's nice and warm. And then the leaves start coming out, right? These bright green leaves on all the trees that were barren during the winter. So I remember walking through the trees, and I love the feeling of the warm sun shining down on me as a kid, walking through the woods. So I think all of us have a certain appreciation for the fact that life on Earth is based upon the energy of the sun, right? The energy of the sun ultimately is what drives all living functions on our planet. And because we are aware of that, we know, right, that there is this deep connection between all living things, between us and plants and animals and all the diversity of different life forms, right? We are somehow connected. And it all starts with the energy from our sun. So let's explore this in a molecular sense. Okay. In general chemistry, and certainly in OChem, you studied what we call redox reactions, right? These are sort of combined oxidation and reduction reactions. So let's revisit these terms so that we just sort of remember what we're talking about when we say oxidation and reduction. So the transfer of electrons from some atom, for example, let's say this atom here, right, atom A, it has 2 electrons that are available to participate in other atomic bonds. So these are electrons that might be, you know, if we think of our example, way at the beginning of the semester, we use sort of an analogy, right, of Val the waitress, who's an electron working in a very busy restaurant, right? And Val, you know, it's a stable job. She's not worried about losing her job, but it requires a lot of energy associated with her running around, right? We said that Val would much rather, if she had her choice, leave that high energy job and take a much more chill or relaxed job, like at the little French bistro that opens up down the street, right? So you think about this atom with this pair of electrons here, right? And the removal of these electrons, right? The removal of those electrons by some other atom, right, is the process of oxidation. So the term oxidation, you know, I think it can be a little confusing because you think like it always has to do with oxygen, right? No, oxidation certainly does not always have to do with oxygen. It's just that on our planet, right, and the way life evolved, oxygen is a readily available atom to participate in reactions where the oxygens take electrons from other atoms, right? So therefore the term oxidation, it's kind of outdated because you have to remember this term oxidation came from, I don't know, like in the 1700s or something like that, right? When chemists were sort of understanding the involvement of oxygen in taking electrons away from other atoms. So they just called this process oxidation. Right? So that atom A that has its electrons removed, it is oxidized, right? So now this A on the right here is in an oxidized state. The other atom, for example, it could be oxygen, atom B, that takes those electrons, right? That is the process of reduction. So by taking those extra electrons, this atom down here becomes reduced, right? And we said, if we are to think about our story of Val the waitress, right, when she goes from a higher energy job to a lower energy job, right, she gains some free time, right? We say now she has some time to do some other stuff and she can go bike riding or take up some other hobbies, things like that, right? That represents the extra energy that has been sort of freed up from going from a higher energy state to a lower energy state, right? Now, Val can use that as her free time to do different types of fun things. But in terms of atoms, when you have the high energy electrons moving to a lower energy state, right, that energy difference is released and it is available for things like the cell to be able to harness it. Right? If there are other molecules available, positioned and designed to take advantage of that. Otherwise, what happens to that energy difference? If the energy difference is not taken up by any other molecules, it is just released, right? It is lost into the environment as heat, right? That's kind of like the leftover energy. So let's look at some just simple chemical examples of oxidation and redox reactions, just so we can appreciate this idea of the difference in energy from start to finish and what that represents, especially if you don't have some way of harnessing it. So here are 2 compounds. Here is potassium permanganate. All right, so potassium permanganate is this compound that's a potassium ion sort of ionically associated with this permanganate ion, right? So it's a manganese with 4 oxygens around it. This oxygen, it has a negative charge to it, right? And then you have this potassium ion with a positive charge. So the way they associate together is sort of like this ionic association. That's why there's like a little dotted line between the potassium and this permanganate. Sort of like when we were talking about sodium chloride, table salt, right? And we said the Na and the Cl, the Na has given up electrons to the Cl. Therefore, the Na has become positively charged. The Cl becomes more negatively charged. They are not sharing electrons anymore, but since the electrons got transferred over, they naturally will stay together because the positive charge of the Na is attracted now to the negative charge of the Cl. We said that's why ionic bonds, like in table salt, are very easily to disrupt, right? When you put the NaCl in water and you have all these little obnoxious water molecules surrounding them, and the water molecules get in between, the water molecules can surround the Cl and the Na, and that's why the salt, the table salt, sodium chloride, comes apart very easily. So this potassium permanganate is kind of like this as well, right? They're sort of loosely associated with each other. If it's just sitting there, yeah, it's pretty stable though, right? But it's very easy to disrupt. Right? So in this case, if you mix in something like a strong reducing agent, like glycerol, right? So glycerol, the 3-carbon chain, is the same glycerol that we've talked about as being sort of that important component of things like our phospholipids. Remember the glycerol, sort of like the staple that holds together fatty acid chains of our phospholipids, and then you have a phosphate group on the 3rd carbon. Glycerol is very useful in that sense because things react with it. It has these hydroxyl groups hanging off. And these hydroxyl groups, you know, I mean, again, they're stable, but they'd much rather, right, be able to be sort of released. Okay. So if you have a strong reducing agent like glycerol and you mix it with a strong oxidizing agent like this potassium permanganate, right, these bonds in each of these molecules so want to break and reform better bonds that spontaneously they will do so, right? And this equation down at the bottom is the equation or the reaction that takes place when you mix the potassium permanganate with the glycerol. Just by mixing them together, right, these bonds start breaking and reforming. And we're not just talking about one set of bonds breaking and reforming. We're talking about a whole series of reactions that spontaneously take place, eventually getting you all the way down very quickly to these compounds down here and the release of carbon dioxide and water, right? In several very spontaneous steps, each step of which, right, represents electrons going from a higher energy state to a lower energy state. And each step of the way, energy is being released, right? That energy difference of the electrons as they go from a higher to lower orbital configuration. Now, if all you have is the potassium permanganate and the glycerol mixed together, then what happens to all that energy? It just gets released as heat, right? And it is a tremendous amount of heat as you go all the way down through these reactions spontaneously, all the way down to like carbon dioxide and water and these compounds here. So this mixture will actually spontaneously start burning and release a lot of heat, right? That represents the difference from start to finish of these electrons in their orbitals. So clearly, the reactions that take place in living cells involving biological molecules, they cannot be this highly exothermic, because right? That's just not compatible with life, right? That's why, I mean, in kind of a nutshell, that's why our living cells don't depend upon glycerol reacting with potassium permanganate, right? Because there's just too much energy that would be released, too much heat, and that's not compatible, right? It's too out of control. Biological molecules, instead, they use the same principle of bonds breaking and reforming, but in carefully coordinated little steps that are manageable or controlled. So with each reaction, there's the release of a little bit of energy, right? And there may be other molecules that are ready to receive that energy to do other types of reactions. So that implies that in biological reactions, from the molecule A reacting with molecule B, right? You have to have some sort of third molecule in there that actually controls that reaction so that it is not just spontaneous. So cellular energy then, all the stuff that's taking place inside of your cells right now, right, is management of these types of biological redox reactions. So unlike the spontaneous combustion of potassium permanganate and glycerol, right, redox reactions in biological systems, they require a little energy being put into them to start the reaction. We can think about that as our cartoon here that we've seen before, that we have this big ball at the top of a hill, and we all sort of intuitively can sense, right, that the ball would rather be at the bottom of the hill because of gravity pulling on it. But it's stable. This ball could sit there at the top of the hill, right, for, I don't know, 10,000 years, right? It requires a little energy to get it going. And once it gets going, then the potential energy that's represented by the ball held high up on the hill, right, wanting to sort of being pulled upon by the force of gravity. Then with a little energy put into it, it can be transformed into kinetic energy and the ball will roll and you can do work. So stable biological molecules in our cells require some energy to start breaking the bonds, right? To be able to encourage them to form better bonds with another molecule, right? And just like we said, with our example of Val working in the sort of high stress, high energy restaurant, it's a stable job. She's not going to get fired, right? Because this restaurant is not going to go out of business. So even though she'd rather leave that job, she tends to stay. And I was kind of joking before. I said, she's like your friend who always complains about her job, but then the moment you say, okay, I'm tired of hearing that, why don't you just leave your job, quit your job, right? Then she's like, no, it's okay, right? So she doesn't go anywhere. It takes a little energy, right, in order to get her motivated to leave that job. And she will only do it if there is a better job to go to. So let's think about it this way in terms of biological molecules. Here is a series of related organic compounds, organic molecules, and they are arranged in a way going from the most reduced state on the left, like this alkane, to the most oxidized state on the right, this carbon dioxide. And remember, the more reduced you are, that means you are more loaded with electrons. The more oxidized you are, it means the more the electrons have been taken away. So you can see here, from alkane, right, you have these two carbons and they're very happily saturated with hydrogens, right? So they are very reduced because they have their full complement of electrons on them. But as you start to oxidize them, right, so for example, if you replace this group here with a hydroxyl group, this represents oxidation because now you have an oxygen that's beginning to take away electrons, pulling the electrons away, right? So this is an alcohol now. If you continue this, you can see you can form a double bond here, representing the oxygen now taking or being more greedy and taking the electrons away from the carbon. So this is an aldehyde. Then over here, you get a carboxylic acid. So now you're beginning to change. You're putting hydroxyl group up here, an oxygen. And then eventually, if you were to cut this bond, you get carbon dioxide, right? Each step of the way representing electrons being kind of like taken away from the carbons. Importantly, this series of molecules, right, this is something that would take place inside of a living cell in your body. It would represent how something like an alkane gets broken down, right, through intermediate steps to eventually lead to something like carbon dioxide. And each one of these steps, you can imagine there'd be some other molecule present here that gives a little bit of the nudge or the push or the motivation for bonds to break. And that molecule would have to be the right shape in order to associate properly with these molecules. Right? And with each step, there is a little bit of energy released. Okay. Eventually, getting down to carbon dioxide, and as far as our cells and our body's tissues are concerned, there's really not much else you can do with carbon dioxide, right? Because that's about as fully oxidized as this carbon can be. Right? And so that's why carbon dioxide is sort of the end result of a lot of metabolic processes. Our body can't really do anything more with it, so we just get rid of it, right? That's why we breathe out carbon dioxide as a waste product. Let's think about how we can quantify this idea of energy, right? Changes in amounts of energy as bonds are breaking, reforming, breaking again, etc. An important concept that's a little bit abstract is this idea of free energy, or Gibbs free energy. That's why it's a G. It's a way of kind of thinking in an abstract way about all the available energy that's contained within the molecules of a biological system at any given time. Right? When I say biological system, I could mean, for example, a single living cell is a biological system, right? So the G would just simply mean, if you think about all the atomic bonds that are involved in all of the molecules in that cell, right, that represents a certain amount of energy. Right? And that would be the G of a cell. Or we can think of our entire body, right? You can say, I want to define a system as just, one human being, right? Because after all, we are a very complex biological system, but we can think of ourselves as a system. That means the G would just mean all of the molecular bonds that make up you at a given moment, and what that represents in terms of the available energy as bonds are being made and broken all the time. So the change in free energy, or delta G, is the gaining or loss of energy in the form of either more complex molecules that are being built up from smaller molecules. So that means you are actually putting energy into smaller molecules to put them together into a bigger molecule, which represents higher potential energy, right? That's almost like putting the big ball back on the top of the hill, pushing it up the hill to get it up there. Or at the same time, right, you have countless examples of reactions taking place in your cells and tissues where bonds are being broken. Complex molecules are being broken back down to simple molecules. And that would represent the release of energy. And that would be like then getting the ball rolling down the hill, and there's the ability to get kinetic energy out of it. So the total change in the G of a system can be thought of pretty intuitively as whatever G is represented at any given moment by all the products that you have from reactions taking place, minus the G of all the reactants that participated in making those products, right? Because that's your change from one state of biological molecules to another state. So let's think about it this way. Let's think about you take, here's a molecule of glucose, right? So we've studied glucose quite a bit. We're going to study it quite a bit more because glucose is a fundamental energy source for living cells. So C6H12O6, right? So there's our glucose. If you think about it, the bonds in this glucose represent energy bonds that can be tapped into, right? If you break those bonds, you can release some of the energy and harness the energy from it. To do that, you would be oxidizing the glucose molecule, right? Breaking it down. And so we said, what's a really great molecule to help oxidize things? Well, it's oxygen, right? So here you have 6 oxygens, or actually 6 O2s, right? 12 oxygens in the form of 6 molecules of O2. So these will be the oxidizing agent, right? And in the process of taking electrons from the glucose, right, the oxygen is itself being reduced. So if we react 1 molecule of glucose with 6 molecules of O2, Ultimately, if all the bonds are broken and rearranged, you will release all that energy that's contained in the glucose, and you're left with, at the end of the day or the end of the reaction, right, 6 carbon dioxides and 6 waters. So the beautiful thing about this, that is both obvious and sort of not obvious at the same time, right, is that every single carbon and hydrogen and oxygen that you see represented down here is present up here. Right? So this represents the full disassembly of the glucose using the oxygen to take electrons away and things being rearranged. So in that process, you will release all of the potential energy that's contained in the glucose, and the change in delta G will be negative 686 kcals per mole. Right? So for 1 mole of glucose, you'll get 686 kcals of energy released. And importantly, right, it's a negative sign. Why? Because the G of the products, the CO2 and H2O, that represents the lowest energy left over in these bonds, compared to the highest energy in this reaction is represented by the bonds when they were in this state over here. Therefore, the G of the products is way lower than the G of the reactants. So if you plug them into this equation here, you realize, oh, yeah, that's why you get a negative delta G, right? So negative delta G means the release of energy. In this photo, I show a marshmallow being burned or combusted, right? And I think we all love to roast marshmallows over a campfire. And, marshmallow is essentially, you can think of it just as glucose, right? And so when you put in energy, so the glucose molecule, right, is stable. So the marshmallow is stable, right? You don't have to worry about your little bag of marshmallows suddenly exploding, right? Because you have a marshmallow, if you hold it in your hand, eventually it's just going to get stale, right? But it's not going to just explode by itself, right? You have to put energy into it in order to get the glucose carbon bonds breaking, right? So of course the campfire easily provides that, right? So the heat from the flame starts breaking the glucose carbon molecules. As long as you have oxygen present in the surrounding air, that supplies the oxidizing agent, right? So there are plenty of places for the electrons to go as they're being the bonds are being broken of the glucose, right? That's why, of course, that combustion, like burning like this, can only take place in the presence of oxygen. Without oxygen present, you don't have something suitable to take the electrons, right? So as the bonds of the carbons in the glucose are being broken, they have nothing else to kind of reform with. So they would just sort of reform with themselves again, right? You wouldn't get it to burn, right? At least with heat, right? And so, of course, eventually your marshmallow catches on fire and it turns all black, right? What's that about? That's because the flame from the external source, right, is so intense. There's just so much energy that you're putting in there. You're putting in way more energy into the marshmallow than is actually needed, right, to get the glucose to fully break down and burn. So in that case, you would drive this reaction even further down to even breaking the carbon dioxides and releasing your, elemental carbon, right, which is your black leftover sad burnt marshmallow, which, a little burnt marshmallow tastes pretty good, actually, right? So importantly, right, in our bodies, in biological systems, you're not overloading the glucose with so much energy, right? So step by step by step by step, it goes down to this last step, carbon dioxide and water, and then that's where it stops, right? And we release those things as waste products of the metabolism of glucose. What if you went the opposite direction? What if you started with carbon dioxide and water? Well, this implies that you could go back up and make glucose from it, right? And after you sort of broke and reassembled these carbon dioxides and waters and made a molecule of glucose, you would then have 6 leftover oxygens, right? That's what certain living things do, things like plants, photosynthetic organisms. They take the carbon dioxide from the air and water from the air, right? And they have to put energy into this to reassemble these complex glucose molecules from these simple carbon dioxide and waters. Where does that energy come from? The energy comes from the sun, right? So the plants, photosynthetic organisms harness the energy of the sun and use that to assemble carbon dioxide and water into complex molecules like carbohydrates, like glucose. How much energy does it take to do that? Well, amazingly, maybe not so amazingly, it takes exactly 686 kcals per mole. Right? It goes into taking the 6 carbon dioxides and 6 waters, breaking all those bonds, rearranging them to get glucose, 1 molecule of glucose, and 6 leftover oxygens, right, that are then released by the plant, because the plant doesn't need the oxygen after it's made its glucose. Importantly, that delta G is positive. And you can see where this is going already, because the right side feeds into the left side feeds into the right side, right? And that gives us our first inkling of when we talk about how all biological functions on Earth, all living things, the photosynthetic and the non-photosynthetic, are codependent. So how do our cells use energy? So let's stop and revisit our definition of energy. What is energy? Right? I've said this before, but all of us have kind of an intuitive sense of what energy is, right? Like, oh, he or she, she has way too much energy. She needs to lay off the caffeine, right? Or he's got way too much energy, right? He's need to kind of chill out, right? But what is energy? The simplest definition of energy that is useful for our discussions of biological chemistry is that energy is the ability to cause matter to move, right? And whatever resulting motion you get of that matter moving. Right? Now, when I say matter, what am I talking about? Well, I'm talking about atoms, right? Because atoms make up matter. So another way of thinking about it is energy is the ability to break and form atomic bonds in molecules, right? Thus causing other reactions to happen. So let's look at one familiar molecule. So here's adenosine triphosphate, right? So we have our adenosine nitrogenous group attached to a ribose sugar. If we only had one phosphate attached to it, we would have adenosine monophosphate, or AMP, which would be one of what? One of the nucleotides that's used to make RNA. Right? If this were a deoxyribose, then it would be one of the nucleotides that make up our DNA. But in this case, this is another great example of how evolution, once evolution and life made use of certain molecules, like adenosine monophosphate to make RNA, right? It uses it for other things as well. So in this case, you add a second and a third phosphate to it. And now what you're doing is you're storing quite a bit of energy in the form of this high energy phosphate bond between the second and third phosphate, right? Why is there a lot of energy represented in there? Because even though the bond is stable, right, this third phosphate group is not going to fall off. Or fall off by itself, I should say, right? The electrons that are engaged in this bond, they're not very happy, right? Because look, these phosphate groups are really bulky. So already the third phosphate group is going to kind of pushed away by the other 2 phosphate groups, right? Because of their charge repulsion and their bulkiness, right? So as a result, there's a lot of energy contained in this bond. So if you were to break a high energy bond like this, right, you would allow for a lower energy bond to form with another molecule. Right? And therefore, that's what we mean by movement, right? The movement of matter, in this case, a phosphate group being moved onto another molecule, right? Or simply the breaking of that third phosphate bond, representing electrons that then want to engage in other lower energy bonds, right? That could be used to actually change, for example, the shape of another molecule. Right. So this is movement on an atomic scale. So there's 6 types of cellular work that require energy. All right, and we'll go over these in the next few slides, but they involve things like the biosynthesis of new molecules from simpler molecules, right? That requires energy. Mechanical work, right? Movement on a macro or a gross scale, like me waving my hands around, right? Concentrating molecules. We've learned about this when we talked about membranes and gradients, how if you move things against a concentration gradient, it requires energy, right? And it's very fitting that we talk about ATP here because we looked at some examples of membrane transporters that use ATP to break that third phosphate bond as a source of energy to be able to force something like a transporter protein in a membrane to change its shape, which would then maybe expel or push some sort of solute against its natural gradient flow. Electrical work, right? So some examples of electrical cellular physiology, managing heat flow, that's very important, right? And bioluminescence. So let's take a quick look at these. We're not going to go in super detail, but I wanted you to think about them. So energy for biosynthesis. Right? So what we're talking about is changes in chemical bonds that take place when a living thing receives energy. Let's think about, for example, a little bean here that's growing, right, into a plant. Right? So as this little plant is growing, it's taking energy from the sun, and it's storing that energy or converting that energy into the making of things like carbohydrates. So once again, you have your water and carbon dioxide, energy from the sun through the process of photosynthesis that we will study a little bit later, right, can combine these to make monosaccharides, like sugars, like glucose. And then you can go a step further, right, and actually chain together these individual glucoses to make something like a polysaccharide, like a starch. Right? So that certainly requires energy in order to do this. Mechanical work, whether you're talking about little, here we have bacteria that have flagella, right, that kind of wave around, and that gets the bacteria to kind of move through, for example, some water, right? Or we have certain structures on cells in our body, like in our respiratory tract, our epithelial lining of our respiratory tract. The cells have all this cilia, right? And this cilia is meant to help actually keep debris from building up inside of our lungs and our airways, right? So these cilia are constantly waving and encouraging a sort of current of mucus to kind of carry stuff out, like dust or pollen, dirt, or even bad things like bacteria or viruses that we breathe in. And of course, mechanical movement, like our skeletal muscles, right? All of these require energy in order to move these different parts of cells around. Energy for concentrating molecules. We said that energy is required anytime you're trying to move a solute against its concentration gradient. Remember, we learned before our last exam that things being moved across membranes is a super important function of living cells. Right? So things can be passively transported, either because they can just diffuse on their own, or they can be facilitated, helped through the membrane by different types of membrane proteins. But no energy is needed as long as those things are flowing along their high to low concentration gradient. Right? Things get trickier if you want to move things in the opposite direction. So for example, if you, in this case here, you have a active transport protein that is going to take solute from one side. For example, these, I don't know what these are, these orange kind of diamond-shaped things, right? It's going to take them from the low concentration side up here and move them to the side where they already have a high concentration, right? And they naturally would not want to do that. So you have to force this protein to change its shape in order to kind of expel, right, or push these orange diamonds from the top part down to the bottom. And so that can be in the form of breaking ATP. So we said that many of these transporters are ATP aces, right? They are proteins that are designed to break that third phosphate group off the ATP and use that energy, right, instead of letting it get lost, right, harness that energy to actually change the shape of the protein. Energy for electrical work, things like building up our membrane potential. So here's another example of an active transporter, a proton pump that uses ATP to move protons, right, from a low concentration to a high concentration. And we said that these types of direct active transport can then be taken advantage of by other transporters that allow, for example, the protons to flow back through from high to low, and at the same time using that energy of the protons flowing through to be able to move some other solute from low to high at the same time, right? That would be our example of indirect active transport. But also things like our nerves, right? Our nerves function by transmitting electrical signals that are, it's not the same as plugging your outlet into the, plugging your laptop into the wall, that kind of electricity, right? We're talking about changes in the membrane potential that are allowed to fluctuate, thereby allowing, right, changes in the membrane potential to propagate or move along the length of a cell. So a really extreme example of that, of bioelectrical work, are things like electric eels or electric fishes. So, I don't know about you, but I always think it's kind of amazing that you have certain organisms that can generate enough electricity in their body to actually be used as a defense mechanism or as a hunting tool, right? So electric eels, they can zap other fish to stun them, and then they can then eat the fish. Right? So if you were to look at the tissues of an electric eel, you would find that these specialized cells, right, these specialized cells that have these, basically these specialized proton channels in them. In a normal resting state, the cell would have sort of your typical membrane potential, right? The inside of the cell tends to be more negatively charged, and then surrounding the cell, right, tends to be more positively charged. But when these cells activate and you have these pumps that start pumping these ions back and forth, these cells are designed to build up sort of a polarity to them, where one side is negative and one side is positive. And if you have enough of these cells lined up and they're all doing this, you end up creating, right, an electrical gradient where then electrons will flow in one direction through the tissue. And then the eel is able to then, right, release that flow of electricity to the surrounding water. Pretty amazing. Each individual cell in the eel that does this can generate about 150 millivolts per cell when it does this. But when you combine hundreds of thousands, if not millions of these in one little square inch of the eel's skin, you can generate something like 100 volts, right? Enough to stun a fish, and certainly enough to stun a person if you end up, unfortunately, wrangling with an electric eel. Heat flow. Heat flow is another very important use of energy. But I want you to keep this in mind, right? Heat is already the natural byproduct of metabolic reactions within the cell. Anytime you're breaking and reforming bonds, going from a higher energy state to a lower, right, there is the potential to release energy. We already said that if you don't have anything to take advantage of that, all of that difference in energy would just be lost to the environment as heat, right? Our cells, right, are much more careful. We have a whole bunch of different pathways involving other molecules that are ready, right, to manage these little reactions, these little step reactions, and take some of that energy to do something else, right? But of course, nothing is 100% efficient. It's not like all of the freed up energy is always used up by our cells. So that's why there's always some energy that is lost as heat. Right? And that loss of heat is important because that's what keeps our body at our homeothermic temperature of 37 degrees, right? 37 degrees Celsius, 98.6 Fahrenheit. Why is that important? Because all of the proteins and all of the biochemical pathways that we evolved, right, are expecting that temperature. The shape of proteins, right, will change depending if they're cold or if they're too hot, right? You threaten to denature the three-dimensional structure of proteins. So the amino acid sequence of the proteins that make up our bodies evolved to be that particular amino acid sequence so that the protein is stable in its three-dimensional shape that it's supposed to be in when it is surrounded by 37 degrees Celsius and 98.6 Fahrenheit, right? As we learned when we learned about PCR, other organisms, right, they don't care about being at 37 degrees Celsius. Organisms like those thermal vent bacteria on the bottom of the oceans, right, those hydrothermal vents, where those bacteria evolved to withstand very high temperatures, right, like 70, 80, 90 degrees Celsius. And so the amino acids in their proteins have evolved to be such, or the order of the amino acids in their proteins are such that their proteins stay in the right shape at 80 degrees Celsius, right? Whereas at 80 degrees Celsius, those same types of proteins in our body would have fallen apart and denatured. So human metabolic processes are optimized for our normal body temperature. But I want you to keep this in mind, right? Because heat is just a byproduct of biochemical reactions, we don't really have any sort of special biological reactions just to make heat, right? One way we can make heat, if we need it, right, is by friction. So the movement of things like muscles, right? The movement of things like muscles generates heat, right? Because then you have a lot of cell activity going on, and you also have movement of tissues against each other. And that's what shivering is, right? So when we're really cold, we shiver so that we can generate more heat. But importantly, that heat is a byproduct of other types of reactions taking place in our cells, right? It's never a direct intended product of biological reactions. And then lastly, bioluminescence, right? So certain deep sea organisms, for example, right, they can generate light. And so obviously that's energy, right? And so that's energy that's coming from biological reactions inside of the cell. All right, let's kind of end with a couple slides, just sort of as a thought exercise here. So if we think of life and living things as being an open system of energy flow, I said if we think of our bodies as being a system, right, that's how we want to define this particular system with like a G, right? And constantly there's a delta G that's changing as we're making and breaking bonds inside of all of our trillions of cells in our body. But it's an open system, right? Because we have energy coming in from the outside in the form of things like the air we breathe, the food we eat. So let's think about this. Here we have a plant stuck in a bottle of water, and this is an open system. It's a living thing, right? It has energy coming in from its surroundings, whether energy from the sun, maybe there are certain nutrients in the water. Certainly, there are things floating around like carbon dioxide, water, other types of gases, right? And through doing its biological functions, there's going to be some energy lost, right, when given off by the plant as heat. If you were to go ahead and put that plant in a closed system, right, I think we all intuitively know that the plant can't survive in a completely closed container like this. Right? But the important thing is, does the plant die right away? No, right? You can close it up in this container, and probably, a week later, you open it up and look, it's still alive, right? Maybe it doesn't look as great as when you started, but it's still alive, right? What does that tell you about the ability of living things like cells to be able to still store energy? So very clearly, right, the plant is able to store enough raw materials, enough energy to at least keep it going for maybe a week, 2 weeks in the pitch black before it really starts withering away once it starts using up all the energy that it has stored. Okay, last slide for today. So what I want you to think about in a much broader sense then, right, I started off this lecture saying, hey, we're going to discuss energy and hopefully impress upon all of us this appreciation for how life on this planet is the interconnection of all living things, starting with the use of energy from the sun. Right? So here we have solar energy shining down on our planet. We have phototrophs, right? Organisms that can take the sun's energy and convert that into chemical energy for their own use. Right? So things that are photosynthetic are certainly phototrophs. Phototrophs can be thought of as a broader category of organisms that can be thought of as autotrophs, right? Meaning that they can feed themselves, right? That's essentially what autotroph means, right? In the process of taking resources from the air around it, the soil, things like that, and using the sun's energy, they can make things like organic compounds, like sugars, right? And numerous other organic compounds, different proteins and things like that, nucleic acids, lipids, things like that as well, right? And in the process, some of that heat is lost, right, into the environment. So then we come along. We is like basically everything else, right? And you're like, I don't see us here. Well, there's a monkey, so that's kind of close enough, right? But everything else, right, that cannot make our own energy, we depend upon taking the energy in chemical form that things like the plants made. So we take the organic compounds and we take that oxygen. Remember, that oxygen was left over when the plants made things like glucose from carbon dioxide and water, right? We take these things and essentially mooch off the plants, right? And we use these as the starting source to power our cells, right? And in the process then of breaking these things down, reassembling these bonds to make complex molecules, we also lose heat, right? Because each step of the way, it's not perfect 100%, right? So solar energy has been converted into chemical energy. And then what do we release? We release the waste products of that, right? Like the carbon dioxide, like the water that was from the breakdown of the sugars. And we release all kinds of other organic compounds as well, like nitrates, right? And these, once again, become the raw materials for the plants to keep this cycle going. Okay. So as we talk more about this, we will talk about the metabolic processes that take place in our cells. We will talk about the metabolic processes that take place in plant cells. But I think this slide is a nice one to get us thinking about how it's all interconnected, right? And we're going to be looking at that interconnection at the level of these biological molecules. Right? Okay, so let's go ahead and stop there.